Hi and welcome back. We're up to the fourth of our videos looking at area of study 2 food chemistry, and this one will look at factors that affect enzyme activity, remembering that enzymes are the first group of proteins that we're looking at in terms of the function within the body. This will cover all the content that we need up until our next SAC, which is coming up next week, which will be an experimental-based SAC looking at enzyme activity and the effect of different factors. So this is critical to understanding that. So let's get started. With enzyme activity, we talked about in the last video how enzymes have a specific three-dimensional shape. That means that they have the formation of an active site, okay, which catalyzes a particular reaction. These structures were stabilized by the interactions between the side chains of the amino acids that form the polypeptide chains of the protein molecule. This means that any loss of shape to that active site can disrupt the activity of the enzyme. So remembering that we have lock and key and induced fit models of substrate binding into the enzyme, if those side chain groups aren't located the way that they are meant to be, the binding interactions between the substrate and the enzyme may not work well, which means that it's not going to catalyze the reaction, it's not going to lower the activation energy, and we won't see the increase in rate of reaction. We define enzyme activity as the amount of substrate converted into the product per unit time. Again, just like kinetics, per unit time is a critical factor in any way that we discuss the rate of turnover in terms of an enzyme. This is dependent on the concentration of the active enzyme. So if we have more enzyme, this means that we can bind more substrate and we can increase the rate of reaction. The concentration of the substrate will also influence this up to the point where all enzyme is bound with the substrate. Okay, so if all the enzyme we have available is bound with substrate, then we hit what we call Vmax, the maximum turnover of that enzyme, because we can't break down any more substrate if the enzyme is already, all the available enzyme is bound up with substrate already. So these two things link kind of like we saw with kinetics previously, where the concentration of the reactants influences the rate of reaction, because as we increase concentration, we will see greater collisions between enzyme and substrate because they still need to collide for the binding of the substrate to the enzyme to occur. But if we have all the enzyme bound up with substrate already, increasing the substrate any further won't increase the rate of reaction. So generally, the enzyme is the limiting factor in these reactions. The other thing that will affect enzyme activity and therefore our rate of reaction or rate of catalysis is the reaction conditions. Because the tertiary structure is based on stabilizing between side chains, if we have reaction conditions that may change the polarity or change the ionization of the side chains or disrupt those intermolecular forces, then we see a change in the active site and therefore loss of activity. So the things that can change the ionization of our substrate of our enzyme is pH. The other thing that can influence our intermolecular forces is, of course, temperature. As we increase the temperature, we're going to increase the kinetic energy. Things are going to wibble and wobble around a little bit more, and our 3D structure may become a little more loose. So any condition that alters the frequency of the binding events between the substrate and the active site will alter the enzyme activity results, and anything that alters the shape of the active site. The diagrams that we see on the right here are what we call enzyme activity plots. What we have is on the y-axis, our dependent variable is enzyme activity. So we would be measuring the product of the catalysis or something like that. And then we can see with temperature, we have an increase in rate of reaction as the temperature increases up to a certain point, where then we see activity drop off rapidly. We can see this mimicked with pH, where depending on the pH that the enzyme is stable in, we will see an increase in activity followed by a drop-off. So there is an optimum pH range that enzymes will work under. There is also an optimal temperature range of which enzymes will function. This is the one we were looking at with substrate concentration. On the x-axis here, we have substrate concentration, so we're increasing the amount of reactant for a fixed amount of enzyme. And we can see initially we have an increase in the rate of reaction up until this is where all the enzyme would be saturated with substrate. So it doesn't matter how much more we add now, we will see no increase in the turnover of product. But we don't see a drop-off because as soon as that substrate is broken down, there is more substrate there to be bound into the active site, keeping the enzyme activity at optimum. So the words we want to use when we talk about enzyme activity are things like optimum conditions for enzyme activity, optimum rate, and things like that. So enzymes, because they are biological, have an optimum conditions under which the activity of the enzyme will be highest. So they will have an optimum temperature and optimum pH range. For biological systems, particularly for human systems, this means that it's around 36 to 37 degrees, which is optimum human body temperature. This is the body temperature that our body is maintained at when we are healthy. If we get below that, we can suffer from hypothermia, where the temperature drops too low for our enzyme activity to maintain healthy function of our body. Or we can have a fever where the temperature gets too high and we see a drop in enzyme activity in our body, which can be dangerous to our health as well. The optimum pH is around 7, usually just above 7 to 8 for most systems. But different parts of our body have different pH, i.e. in the stomach, the enzymes that operate in our stomach will operate at pHs of 1 to 2, highly acidic. Our small intestine, the pH is around 8 to 9, so slightly basic. So dependent on the purpose of the enzyme, where in the body it's located, the optimum pH can change. With temperature, it's a standard kinetic curve initially where we see an increase in temperature, rising to the optimal temperature, and then the drop-off after. With pH, the pH which an enzyme activity is greatest is known as the enzyme's optimum pH. We can see that by finding the maximum of the curve if we plot the activity. As I mentioned before, this can differ depending on where in the digestive process the enzyme is active, because enzymes, remember, catalyze the breakdown of different molecules primarily, so they're used in digestion. Pepsin, which breaks down proteins, is active in our stomach, and this is breaking large peptide chains into smaller peptide chains. And its pH, its optimum, is approximately 1.5. This is because pepsin is found in the stomach, which has a highly acidic environment. Amylase, on the other hand, salivary amylase, which tells you it's found in our mouth, in our saliva, is responsible for starting to break down carbohydrates, starch. This occurs in the mechanical digestion in our mouth and then is promoted by the amylase in our saliva. This breaks larger carbohydrate molecules down into smaller disaccharides, which are then processed further on in our body again. But the digestion process starts in our mouth, and the pH of our mouth is close to 7, and we can see that the optimal pH for amylase is optimized for the environment we find it in, which is about 7.2. If amylase gets into our stomach when we swallow our saliva, it will be broken down in our stomach by pepsin. Okay, so amylase will not continue to work in our stomach because it will be outside of its optimal pH range. And we can see the activity of the amylase here drops right off. So in the stomach, we would expect amylase to be completely broken down and not active anymore. With changing pH, what we have is that the overall three-dimensional shape of the enzyme is changed. Because it alters the charge on some of the amino acid side chains, those interactions are no longer present to stabilize the specific active site. So changes to the shape can prevent or reduce the binding of the substrate, therefore reducing the activity of the enzyme. Extreme changes in pH will have a large impact on the stabilization of the overall structure, and we get what we call enzyme denaturing. With temperature, we see a similar effect, except at lower temperatures, the enzyme activity is lower due to simple collision theory and kinetics considerations. At lower temperatures, we have lower collisions, we have less collisions, therefore we have less successful collisions, so we have less binding of the substrate into the enzyme. As the temperature increases to the optimum, we will see an increase in activity. Above that temperature, we see destabilization of the active site and eventually denaturation of the protein, i.e. the protein unraveling entirely and us losing all function of the enzyme. So we see less activity at low temperatures due to kinetic theory. We increase the temperature, we increase the number of collisions, therefore increasing the chance of successful collisions up to our optimum temperature. Above the optimum temperature, we start to see loss of shape of the active site and therefore loss of activity, until we reach too high a temperature that now the overall shape of the protein is disrupted and we see what we call denaturation of the protein and loss of all activity. The term denaturation refers to the process where we disrupt the tertiary structure and therefore lose the biological activity of the protein. This can come about due to a change in pH, an increase in temperature, or an addition of certain chemicals that can disrupt the intermolecular forces stabilizing tertiary structure. This can be salts or heavy metals. Okay, so our functional protein, remember, has a specific three-dimensional shape. As we alter the side chain interactions, we alter the stabilization of that tertiary structure. We still maintain some of our secondary structures, but it's not forming the overall three-dimensional shape. As we add more things to disrupt this, we even lose our secondary structures. So these changes of conditions cause the protein to unfold, which results in loss of structure. And then if it unfolds far enough, what we get is a tangling of those protein strands, sort of like knotting up pieces of string, and we lose all function permanently. So permanent denaturation, we have the active protein, it's denatured, the influence on this causes tangling of the strands, and we get coagulation. Okay, so we have denaturation followed by coagulation, and to coagulate means to clump together. Okay, so the protein strands are getting knotted and clumped together. Those knots, essentially, the tangling of the strands is so bad that you get clumping and usually a solid forming. So when we go from raw egg to frying an egg, as the whites start to go from clear to white and we get hardening of the egg yolk and white itself, that is because the proteins are starting to coagulate and clump in an irreversible way. If we were to look at milk, if we warm milk, we are inactivating the protein, but if we allow it to cool, if we don't take it up too high, the protein will refold. So we can warm and cool, and those intermolecular forces will restabilize the secondary and tertiary structures. However, if we take it too far, what we start to do is split the proteins from the watery matrix. And this is what we see when we make cheese, when we change pH. So coagulation, as we said, for denaturation, we disrupt the intermolecular forces within the chain, disrupting the three-dimensional shape. But then what we do is we start to form new crosslinks and tangle these strands together in such a way that it is irreversible. This could be cysteine residues that are not usually close together, getting close together, forming disulfide bonds. and the formation of other crosslinks that would not happen at lower temperatures. This prevents the protein from returning to its original shape when that stimulus is removed, i.e. when the temperature is cooled, okay? But we cannot get it back to the original shape. So the protein, the enzyme, its function will now be completely destroyed. Denaturation is where we are changing the intermolecular forces of the tertiary and secondary structures. It does not break the primary structure of the peptide. If we want to break primary structure, we need hydrolysis through digestion to actually break the covalent bonds within the peptide backbone. So it's important to remember that with denaturation, we are looking at the loss of tertiary structure, sometimes secondary structure if we are at the point of coagulation, but it will never change the primary structure of the peptide. Once we change the primary structure, we have actually completely destroyed that, and that's what we see when we burn things and they start to turn black and result in carbon. In order to determine whether or not we are seeing a loss of activity of the enzyme, we can run what we call an enzyme assay. This is an experiment to determine enzyme activity. Usually we will use some kind of indicator to detect the presence of the product that we're after. If it produces a gas, we can measure that, or a change in pH. We can monitor the color using UV-Vis, spectroscopy. We can use all the other types of things that we did with rate of reaction in order to measure this, because essentially we're measuring the effect of a catalyst on the rate of reaction. Let's have a look at a VCAR question. So this is a simple multiple choice that you will see typical of enzymes and their activity. This one asks us, which one of the following is not true of protein denaturation? So when we're reading this, we need to make sure that we know what's not true. We need to remember that denaturation is a loss of function due to the loss of three-dimensional shape of the tertiary structure, so it is side chain interactions only. So once we're primed with that information, we can look at the options. It could result from a temperature change. Yes, we know that is the case. It can be caused by a pH change. Yes, it can also be caused by the presence of salts and metal ions. It alters the primary structure. No, denaturation never breaks the covalent bonds within the protein backbone, so it doesn't change the sequence. It results in the change of the shape of protein. Yes, meaning that we were looking for the one that is not true, so the answer must be C. Looking at a short answer question, we see here that we have a number of different ones that link to amino acids as well as proteins. So we're given the fact that there are 4 optical isomers of ascorbic acid. Only one of these, L-ascorbic acid, vitamin C, is active in the human body. So this is telling us that to remember that amino acids are optically active, they can have enantiomers, they are chiral, and therefore they interact differently with other chiral compounds. Vitamin C is essential for the human diet and has many functions. One of its functions is as a coenzyme in the production of the protein collagen. Coenzymes, they're working together with enzymes. These are the ones that need to bind in order to facilitate the enzyme activity. So during the synthesis of collagen, vitamin C acts as an electron donor. It's telling us what it's doing. Vitamin C needs to be there to donate electrons, and collagen is important in the body. A lack of collagen results in a condition called scurvy. So we know that vitamin C is acting as a coenzyme and that its chirality matters. The second most common amino acid in collagen is proline. Draw the zwitterion of proline. Okay, I want you to pause this and have a go at answering it yourself, remembering that proline is our unusual amino acid where the side chain forms that ring in the backbone. Okay, remember that. The next one says to describe the role of vitamin C as a coenzyme in collagen synthesis. Use the physical and chemical interactions of vitamin C with the enzyme to explain how the enzyme is able to catalyze the production of collagen. So, in this question, when you put together your answer, you want to look at the fact that it's 3 marks. It has asked us to describe the role of vitamin C as a coenzyme. Okay, so if it is acting as an electron donor, it is a reducing agent. Okay, so we're going to discuss that. That is its chemical, because it is facilitating a reduction reaction. And physical interactions, remember that the coenzyme needs to bind to the enzyme in order to prime it to carry out its function. And therefore, we're going to talk about how that will then allow it to catalyze the production of collagen. So you're going to talk about how enzymes do that. Pause the video, have a go at answering it, and then come back and look at the mark scheme. Okay, hopefully you have had a go at answering this question and the following two that were on the next slide. So let's have a look. As I said, this is our structure of protein. And you can see here that 72% of students in the state got this wrong. So this was our proline, and it was asked to be the zwitterion. So they were looking for two things. One, the correct ionization, the loss of the hydrogen on the carboxyl, and the gain of the hydrogen on the amino part. So with proline, the way it's drawn in the data booklet, this is our carboxyl through to amino, making this one in here our alpha carbon. There's going to be a hydrogen off here. Okay, so that gives us our 4 bonds, which means we need to lose the hydrogen from here to form the carboxylate anion, gain a hydrogen on here to give the positive on the nitrogen, even though it is part of the ring. Then we just draw the ring straight from the data booklet. So you needed to convert it to its zwitterion, and then a lot of people had trouble drawing the structure from the data booklet. So I would recommend that you make sure you can draw the zwitterion of the trickier amino acids. With the vitamin C question, again, you can see only 13% of people in the state were able to attain the third mark. 27% of people got 2, and 24% of people got 1, with 36% of people in the state getting 0 marks for this. This was generally not answered well because students didn't break it down according to the chemical, physical, and enzyme activities parts of the question that it required. In order to get the marks for this, you needed to state that vitamin C binds to the surface of the inactive enzyme and changes the shape of the active site. That was the role of a coenzyme. Okay, so it primes the coenzyme, it activates the enzyme, ready for binding of the substrate. The second point is that the substrate is now able to attach to the active site and the reaction can be catalyzed. So the binding of the coenzyme activates the active site, therefore allowing the substrate to react and catalyze the reaction. From there, we then needed to talk about the chemical role of the ascorbic acid in the redox reaction. So the ascorbic acid is oxidized during the reaction because it is a reducing agent. Okay, it was giving up electrons, so it was reducing something else. It was acting as a reductant. You needed to refer to both physical and chemical and the enzyme, which means that you needed to state what things in the question meant. Simply restating electron donor did not give you the mark, okay? And you needed to point out that the coenzyme binding to the enzyme active site primes it, makes it ready for the substrate to be able to bind. Without its presence, the enzyme is not active and is unable to catalyze the reaction. And you'll note here that it says an annotated diagram might have been effective. So if you're finding you can't find the words to properly explain what you want, but you can draw your enzyme binding diagrams, that's a great place to go when answering these questions. The other question 4C is relating to optical isomers. So you had to give any definition of an optical isomer. So 2 or more forms of the compound that have the same structure but are mirror images must contain a chiral carbon. Okay, so you needed to make sure that you were talking about what makes something an optical isomer and that idea of that mirror image. So again, this was answered, 48% got the mark for this one. 52% got it wrong, largely because they couldn't be specific enough in what they meant in trying to get the right words. So some students missed the isomer being optical and confused that with geometric in terms of cis and trans. Okay, it is important that we know optical isomers are referring to our enantiomers that require a chiral carbon. The last part of this question was again referring to coenzymes. This we will cover more when we talk about vitamins as well. Okay, that's it. Of course, we're going to practice doing some VCAR questions in class. Make sure you've made note of anything that you're confused on or you need help in terms of structuring those sentences and phrasing. And I will see you in class.